Hello again, and welcome to the November AJP issue of Heart and Circulatory Physiology's video table of contents, or from the editor's desk. As you can probably tell by the background, I'm not in my office this month. I'm at the American Heart Association meeting in Orlando, so it's a timely place for me to make this report. This month is truly spectacular. In the November 1st and 15th issues, we've published 4 review articles, 1 editorial focus articles, and 31 original research papers. It's been really hard to choose which to highlight, but here's a small sample. Matsushima and Sarushima provide a review of the role of Sirtuins in cardiac disease. They discuss how these class 3 histone deacetylases are linked to longevity and DNA repair, as well as mitochondrial biogenesis and circadian clocks. The protective effects of various Circs is discussed in detail. Anyone interested in longevity and cell survival should really read this review. Schwartzall et al. Describe a porcine model of hypertension and hyperlipidemia in order to study diastolic heart failure. Hypertrophy and dilation were produced without a reduction in ejection fraction. The left ventricular pressure-volume relationship was shifted to the left. Left ventricular superoxide production was increased, in part attributable to NOS uncoupling. A large animal model of HFpEF should be beneficial to those investigating the mechanisms and targets of diastolic dysfunction. Whistler and co-workers investigated whether beta-arrestin 2 contributed to thoracic aortic aneurysm formation in a mouse model of Marfan syndrome. siRNA targeting beta-arrestin 2 inhibited angiotensin-stimulated expression of pro-aneurysmal signaling mediators in primary aortic root smooth muscle cells, such as MMP2 and 9, which was inhibited by blockade of ERK1/2 or the EGF receptor. These results suggest that beta-arrestin 2 contributes to thoracic aortic aneurysmal formation in Marfan syndrome by regulating ERK1/2 dependent expression of pro-aneurysmal genes and proteins downstream of the AT1A receptor. Azam et al. Studied the effects of dichloroacetate, a glucose oxidation stimulator, on cardiac contractile dysfunction following ischemia-induced ventricular fibrillation in a Langendorf perfused preparation. Pretreatment with DSA improved post-ventricular fibrillation, left ventricular developed pressure, and DPDT max. Lactate and pyruvate dehydrogenase phosphorylation were significantly reduced. The presence of exogenous fatty acids did not affect the beneficial effects of DCA. The authors conclude that enhancement of PDH activation by DCA mitigates contractile dysfunction following ischemia-induced ventricular fibrillation. Singh and colleagues evaluated the vascular repair mechanisms of the ACE angiotensin II AT1 receptor pathway versus the ACE2 Ang1/7 mast receptor pathway in human CD34 positive cells. Migration of CD34 positive cells was enhanced by Ang1/7. In the presence of ACE2 activators, proliferation was enhanced, and this was blocked by DX600, an ACE2 antagonist. Ang2 treatment prior to activation of ACE2 attenuated these responses. The attenuation was reversed by apocyanine, implicating a ROS pathway. These results suggest that the ACE2 pathway stimulates functions of CD34 positive cells, whereas Ang2 attenuates these functions by acting on blood mononuclear cells. Finally, Chi et al. Studied the role of store-operated calcium channels on the migration of human c-KIT positive cardiac progenitor cells. They found that store-operated calcium channels mediated calcium influx, and TRPC1, STIM1, and ORI1 were involved in formation of these channels. Silencing TRPC1, STIM1, or ORI1 with siRNA significantly reduced calcium signaling and decreased cell proliferation and migration. This novel finding may be important in developing strategies to enhance cardiac progenitor cell migration. Well, that's it for this month. Remember, we'll be posting videos once a month and podcasts as they occur. So check our YouTube channel and our AJP website. I'll see you next month, and please don't forget to send us your best work now.